I'm interested in bacterial anatomy, so I'm both a microbiologist and a cell biologist. And the question that we're primarily interested in is how these very small organisms are organized, all the way down from the scale of individual proteins out to the whole organism. So we'll start out by putting the cells onto a flat surface, focusing the objective lens so that we can see these bacteria and the anatomical features within them. And then we ask the microscope to take what's called a time-lapse video image. So we take a picture every few minutes and we watch how those anatomical features change locations over the course of the cell cycle as the cells grow and divide. All of these bacterial species, whether they're pathogenic or non-pathogenic, share a common trait, which is that they're highly organized. And there's one particular protein that the lab tends to focus on called PopZ, or polar organizing protein PopZ. And that protein is localized to one extreme end of the cell called a cell pole, and it helps to organize the cell. It pulls in other proteins so that they co-localize the same end. Proteins like PopZ are used for cell organization, not just in alpha proteobacteria, but in all forms of life, including humans. Altogether, we're studying maybe 5 or 6 different species, all of which have a different and unique amino acid sequence of PopZ. And what we'd like to learn is how the binding partners of PopZ are changing or how they're different in these different species. In other words, how does the amino acid sequence of PopZ change in between these species to allow new binding partners and new cellular functions? The traditional view of how a protein interacts with another protein is it has this structured interface where we could describe the three-dimensional shape of the outside of this protein and some of the chemical features of the individual amino acids that are on the surface and sort of get a view of the three-dimensional surface and in that way describe the binding interface. One of the major focuses of the lab is the structure and function of this polar organizing protein PopZ. The general term that describes the structure of a protein like this is intrinsic disorder, which means that it has no structure in and of itself, but will undergo a distinct disorder, that is structural disorder to structural order transition, as it interacts with each one of its 10 different binding partners in the cell. A curious feature about PopZ is that it has sort of 2 different halves of the protein. One of the halves allows PopZ to interact with itself, so you have a network of interactions among multiple PopZ proteins that forms this large 3-dimensional structure. The other half of this protein is the part that's intrinsically disordered, and it's this intrinsically disordered part of the protein that has this interesting ability to interact with not PopZ, but many other proteins, so up to 12 different proteins. If you compare those 12 proteins, they actually have nothing in common. There's no unique feature in any one of those 12 binding partners that would act as an interface for PopZ. The questions that led to the current theme of the lab were prompted by trying to understand how this one part can interact with 12 different parts. We're starting to understand how it's not 12 different interfaces, but it's one disordered protein which undergoes a distinct disordered order structural transition for every one of its different binding partners. Disordered proteins are present in all kingdoms of life, and a general and unanswered question in the field is exactly how their disorder correlates with their functions of having this ability to bind to many other proteins. We're able to bring to bear the power of rapid genetics, easy biochemistry, and fast and easy experiments that tell us what it's binding to and how strongly. I think the primary goal of the lab and my own interests as a biologist are related to basic principles of how life works. What I hope is that understanding how this works at a very basic biochemical/biophysical level will have lessons that can be applied to other proteins that have similar functions. So there are 2 bacterial proteins that have been shown to have this quality of intrinsic disorder and the ability to interact with multiple proteins. The suspicion is that there are many others out there, or several others. Which proteins those are and how they contribute to cell organization could be an important and fruitful question in future years.